Hi everyone, this is Jim Brase. I'm Deputy Associate Director for Computing at the lab. I'm going to talk to you today about our laboratory R&D response to the COVID-19 pandemic. Computing and the Computing Directorate is at the heart of what we're doing here. Our ability to do precision measurement in biology, to create lots of new data, to then build computational models around that is really changing everything in this area. It's changing biology and medicine. It's changing what we can do in biodefense and everything around the bioeconomy. And it's really at the core of our response on COVID-19. We've been working in the area of multiscale learning models that bring together computational models based on theory with experimental data in an AI-driven machine learning framework for some time. And we're applying this all over the lab, from the stockpile program to ICF and to biological systems like these. We have projects that we're doing with the National Cancer Institute to build predictive models for cancer. We have a partnership called ATOM with GlaxoSmithKline, the big pharma company, and National Cancer Institute and UC San Francisco to accelerate drug design using these types of models. Programs with the Department of Defense for rapid design of vaccines and antibodies, with the American Heart Association for new types of drug safety models. When COVID-19 hit us, we brought capabilities from all of these projects together to create a program of really trying to respond and accelerate the development of new therapeutics for this pandemic. And this hasn't been done just at Livermore. It's happened across all the national labs. We've all come together and worked together in the National Virtual Biotechnology Lab, which is how we actually organize ourselves in not just in therapeutic response, but in a wide variety of areas responding to the pandemic. Computing and the computing director is really central to this, to both the leadership and the execution of all this R&D. So we're applying high-performance computing in a number of ways to COVID-19 response. We're developing new types of therapeutics, whether small molecules or antibodies that can actually bind to the virus and to proteins within the virus and change its operation and prevent it from infecting human cells. That's incredibly computationally intensive. So we've worked with DOE and NSA and some industry partners to expand the computing capabilities that can be applied to COVID-19, and I'll describe a little of that. We're also working in a broad consortium all across the country with DOE, NSF, NASA, and a number of companies to provide computing cycles to research groups all around the world working on COVID-19. And I'll describe some of that activity as well. When we develop a small molecule therapy for a virus like SARS-CoV-2, the general approach is that we start with very large sets of molecules that we think might be and have the right form to interact with the virus. We then do computational screening of those molecules at several stages to basically narrow it down to compounds which are predicted to have good binding strength to viral proteins. But even at that point then, you need more than just binding to the virus to be a good medicine. It also has to be safe. It can't bind to other things in your body, heart proteins, liver proteins, and so on. It has to have good pharmacokinetic properties. That means that the molecule can actually be transported through the blood to where it needs to go. It can last long enough in your body to do its job. So that set of molecules actually goes into a molecular optimization loop where we're predicting those properties with machine learning models, comparing them to design criteria, and then in an optimizer, tweaking them just a little bit to try to make them closer to the desired properties. And then we go around that gray optimization loop in the figure thousands of times, modifying those molecules until they get closer and closer to what we want. Then we'll take out the best ones and we'll run experimental assays on efficacy and safety and PK to test and validate that we actually have gotten what we think. So we've been running this process now for several months. We've identified 4 lead compounds as potential SARS-CoV-2 spike antivirals. The spike is the protein that you see sticking out from the spherical virus. It's how the virus actually enters human cells. And we're demonstrating on one of those molecules real actual interference with pseudoviruses, weakened viruses, in capability to cause infection. So these molecules are not there as real medicines yet, but they're moving rapidly along that path. We're doing the same thing in the design of antibodies. We're using computation to predict the properties of antibody designs, and then testing those experimentally and iterating the design cycles using the same kind of optimization structure that I showed with small molecules. As I said, this is incredibly computationally intensive. To basically discover one new binding antibody here, we've done over 1 million simulations of these large protein interactions with many millions of HPC core hours spent on this. But you can see we're actually being successful. The data on the right side at the top shows that as we increase the antibody concentration along the x-axis, we see decreasing viral activity, and that's shown in the plot. That's exactly what we're looking for. This isn't there yet. Again, we need another round of optimization on these to get that down even further, but this is looking very promising. And we're working hard with our partners on this project in the Department of Defense to move this antibody along rapidly into the process for further testing. So as I said, this is very computationally intensive to run these simulations. So working with DOE and NNSA on investments that have come in through the CARES Act funding, and with co-investment from our partners at Intel and AMD, we've really helped to substantially grow our computing resources that can be applied to these problems. Corona is one of our main clusters in our open computing facility. We've been able to really boost the performance of that system up to over 10 petaflops based on this funding. And it's a real workhorse for these simulations that we're doing. Ruby is a new system that's coming in now, which will provide CPU cycles and supplement quartz in our capability in that area. We're also investing in new storage systems, in new data distribution systems. And these investments that we've been able to make for COVID-19 have really boosted the open computing capability at Livermore substantially, and that will ultimately benefit everyone at the lab. Finally, I want to describe the work we've been doing in the COVID-19 HPC Consortium. This is a group of high-performance computing providers. DOE and many of the national labs are involved in this. The NSF computing centers are involved, NASA computing, and then a number of universities and many companies led by IBM, but with strong participation from Microsoft, Amazon, Google, HPE, and many others. We've actually provided computing capacity now to over 80 projects all around the world, and with projects that range from understanding the basic structure and function of the virus to design of new therapeutics along the lines of some of the projects I've talked about, to designing new interactions and new methods for modeling patients and epidemiological models and such. So there's a wide variety of work going on that's having real impact on the response to the pandemic. So the work that the Computing Directorate and the laboratory is doing on COVID-19 is an important part of our overall response in DOE and NNSA to the virus. It's making important contributions for the country, and it's really helping us to establish new capabilities to respond not only to this pandemic, but to future pandemics that may happen. Thanks very much.